Hi, my name is Jay Sugarman, and I want to welcome you to Innovation Showcase. The main purpose of this ongoing series is to inform viewers about exciting innovations and creative individuals across the fields of business, science, technology, education, and the arts. Today, via Zoom, we're fortunate to have as our guest Dr. Dan Distel. Dan is the director of the Northeastern Ocean Genome Legacy Center, which is located in Nahant. The center is an international leader in collecting, preserving, and sharing the DNA of marine organisms, creating a living archive that supports scientific discovery, biodiversity conservation, and future innovation. During the program, Dan will be sharing the history and mission of the center and how it's grown, developed, and operates today. In the process, we'll come to better understand and appreciate why preserving ocean genomes matters and how this work could shape the future of research, medicine, environmental resilience. Let's start by meeting Dan and then finding out all about the Northeastern Ocean Genome Legacy Center. Welcome, Dan. So delighted to be able to be here.
Well, thank you for inviting me.
It's been fascinating reading about the center, eager to find out more and have you inform viewers. But before we jump right to that, I think it would be of interest if you briefly share a little bit more about your background, perhaps academic, professional interest, and what your current position entails, please.
Sure. So I am currently a research professor at Northeastern University, and I'm the director of the Ocean Genome Legacy Center. Um, so the Ocean Genome Legacy Center, we call it OGL, um, has just completed its 20th anniversary year. So we've
Mm,
been around for a little while,
wow, yeah.
and it has an interesting history. Uh, we began, as I said, about 20 years ago with an effort by a very interesting guy named Don Combe. Don was the founder of New England Biolabs, which is one of the first biotechnology companies in the world.
Mm,
And Don was a very interesting character. We lost him a couple of years ago. Um, but he had the idea of creating this biological repository um, to preserve DNA from marine organisms all around
mm,
the world. His inspiration came from an article in Science, which was an editorial uh, authored by several of his friends that asked the question, we know that we're losing organisms on our planet from various human activities at an astonishing rate.
yeah.
Um, why aren't we saving DNA from some of these organisms? We know how to do it. It doesn't require any new technology. It's not
Mm,
terribly expensive to do.
mm,
And really, DNA is the most informative part of an organism you can preserve. You can preserve all kinds of things, tissue, a critter in alcohol, photographs, audio and video recordings. But an organism's DNA is really an incredible record of its history, its biology. It
mm.
tells us a tremendous amount about that organism, how it relates to other organisms on the planet. Um, so why aren't we preserving that? And Don read that article and said, well, this is something I could do something about. And he put down the initial funds um, to create Ocean Genome Legacy. He then hired me as the director
Mm.
and gave me free rein. Just said, here's the
Wow.
general idea. Do something, do something great.
Fabulous.
Which was a tremendous thing. That was a great opportunity. And so what we decided to do was build a very diverse collection that's wide open. Anyone can access it. We wanted to be diverse in terms of taxonomy, and the different types of organisms that we collected. We wanted to be diverse in terms of geographic localities that we collect from. And we wanted to be diverse in terms of the sort of environments or ecological features of the places where we were collecting. Um, and so, Don gave us free rein to do that. Um, the only exception being that he really wanted to be marine focused because we know so little about the marine environment compared to
Hmm.
the terrestrial world.
How are samples collected, preserved, and protected for long-term use?
That's a great question, and it very much depends on the type of sample.
Mhm.
Um, but um, there are a variety of ways to preserve materials. Cryopreservation is generally considered kind of the gold standard
Mhm.
of preserving DNA in tissue samples. But there are a variety of other ways that it can be done. It can be done chemically. There are various chemical preservatives. The most commonly used preservative is ethanol, right?
Mm.
Alcohol that
Mhm.
you can probably find in one of your closets somewhere, I'm sure. Uh, and ethanol has been historically the preservative of choice going back hundreds of years, if not more. Um, but it's good at preserving the morphology or the appearance of samples, but it's not very good at preserving the quality of the DNA inside.
Mm.
So that's actually an area we've worked quite a bit on. We have a project that we're very proud of called Student Research in DNA Preservation. And that's been going on probably about 15 years now, where we employ students to work on long-term DNA preservation projects where we test different types of preservatives. Um, and it's actually been a very successful project. We have had three publications come from that, all three of them
Nice.
with undergraduate students as lead authors.
Fabulous.
And our students have done fantastic work. Um, and what I'm most proud of is the fact that we were able to explore different preservatives that are currently being used and to actually discover a new chemical preservative. It's not new to science. I mean, people have been using it to preserve food and purified DNA samples for a long time. It's called EDTA. Um, and you might find it if you look on packaging of some
Mm.
cosmetics or foods. Uh, it's used as a preservative, but people hadn't recognized that it could also be used as a preservative for whole bits of tissue. Um, so the way we discovered it was we were looking at a preservative that had been developed in the 1990s
Mhm.
that's becoming quite popular because it's inexpensive, it is safe, and it works at room temperature. Um, but it has three ingredients. One of them was EDTA. The other two ingredients, one was called DMSO, it's another chemical,
Mhm.
and table salt.
Mm.
So we decided to ask our students to deconstruct this preservative and see how each of the components works on its own, and then look at all the pairwise comparisons that we can make, all the pairwise combinations, and then compare that to ethanol, the most common preservative. And what we found was even though most people, they call it DMSO salt or DMSO based preservatives, it turns out that DMSO has no preservative effect at all. In fact, in many cases, it makes the preservative worse.
Mm.
And salt also has little contribution. It's the EDTA, the part that no one ever mentions,
Ah.
that is actually the active ingredient. And once we knew that, um, our students were able to look at the um, chemistry, the known chemistry of EDTA, and figure out how to make it work better. And so the first paper was the deconstructing uh, uh, it's called DESS, that preservative. So the first paper was called Deconstructing DESS. The second paper, we took the chemistry of EDTA and we figured out that by changing the pH of the solution, we could actually make it much more effective. Uh, and then we followed up with a third paper, which showed another kind of application for the EDTA preservative, and that is that often when you freeze, freezing is the gold standard. I mentioned that before. But the problem with freezing is to get the DNA out, you have to thaw the sample,
Hmm.
right? And even if that thawing period is very brief, a matter of seconds, DNA can begin to break down. So what we discovered was that by putting frozen samples while they're still frozen into an EDTA solution and letting them thaw in that solution, you could eliminate that damage that happens during the degradation. And this turned out to be very important, or we think it's very important, because there are millions of samples in freezers all around the world
Hmm.
that people might try to use a bit of it and say, oh, we didn't get good DNA. But we found that if you take that very same sample, another piece of it out of the freezer, thaw it in EDTA, you get great DNA.
Fabulous,
So
fabulous.
it's a way to get to add value to all of these samples that are in freezers all throughout the world.
Terrific, terrific. And just another example, as you were sharing that and the student involvement of the incredible way Northeastern introduces students to the real world, whether it's through world-renowned co-op program or other opportunities like the one at the center. How do scientists around the world access and use the genetic material that's stored at the center?
That's another great question. So for our first roughly 18, 19 years or so, we had a homemade database that we built ourselves. Um, and we gave people access to our basically our taxonomic list
Mm,
via our website. And that was useful, but we realized that we wanted to be able to share more of the information than just a list of which organisms we have in our
mhm,
collection, because we collect a lot of different kinds of information about each organism. We collect we record the collection location, who collected it, how it was identified, various aspects of size, shape, what part of the organism was collected, how it was preserved, and so
mm,
on. That's all valuable information to researchers, but we weren't able to share that. So we joined a consortium, and it's called the Arctos. Let's see, I never can remember the full name. Collaborative Collection System, I think it's called.
mhm,
Basically, it's a group of about 100 different collections around the world who have joined together in a collaborative effort to build a database that can accommodate all kinds of information. So this isn't just biological collections. It's also like uh, cultural collections, book collections, all kinds of collections. So the big challenge was to find a way that you could build a database that could serve all their needs.
mm,
It's a huge challenge. It's ongoing, but that's what we liked about it was that we could have input ourselves into how the collection management system grows, um, and what kinds of information we can access and how we can make that information available to the
mhm.
public.
What are some discoveries or breakthroughs that have already come from the center's collection?
There's been quite a number of them. So so one of the reasons we wanted to provide more information was, the collection location information is very valuable because
Mm,
it helps researchers track where a particular species can be found around the planet. So we share that information with an organization called GBIF, which is the Global Biological Information System, I think it is. um, and also another one called OBIS, which is the Ocean biological biogeographic information system.
Mhm.
Um, those organizations then track who uses the data that
Ah,
we share with them so we can get access to that information. And in that way, we've actually found that our data is being shared um, tens of thousands of times. Millions of records
wow.
are being shared each year. And we can also track what publications use that data. So we know that to date, we've tracked over 400, probably closing in on 500 publications
Wow.
have used information from our collection. That's really satisfying.
Hmm.
And then we know through our collaborations how some of our samples have been used. So, for example, we have a very active collaboration with a group of medicinal chemists at University of Utah, and now
Hmm.
one of them has moved to University of Rhode Island, and another one is at University of Washington. Um, so through these collaborators, we've shared materials with them. They've used those materials to search for potential drug lead compounds. And so
Mm.
a couple of really interesting ones have come out of it. Several have come out of it, but two are really worth mentioning. Um, One is called Turner cyclomycin, uh, named after Ruth Turner, who is now deceased, but was an expert in a type of marine bivalve, which I happen to do a lot of work with, called
Mm.
shipworms.
Hmm.
Um, and Turner cyclomycin turns out to be very effective against a bacterium called Acinetobacter baumannii. It's a very important multiple drug resistant, often hospital acquired infection. Uh, it's one of highest concern because the medical world is down to really just one antibiotic that's effective against it. If they lose
Wow.
that antibiotic, then there's no defense.
Mm.
So this is a very promising new antibiotic. Um, our colleagues at Utah are working on how does it work? What is its mechanism of action? And can it become an antibiotic for human use? Another one came from a former member of our research group who is now faculty at, I believe it's University of Washington. I
Hmm.
probably should double check that. Um, or is it Washington State? At any rate, um, she looked at another drug that was first identified as an antibacterial. She looked at it as a potential antiparasitic drug. And it turns
Mm.
out that it's quite effective against a group of parasites called apicomplexan parasites, of which malaria, the malaria parasite, is
Mm.
one. And it turns out that it is effective against all stages of the malaria parasite. So these are two really potentially impactful applications of materials from our collections. Other kinds of things people have used it for. Um, People have used materials from our collections to develop a method of identifying seafood. Um,
Mm.
so you get a fillet of fish in the supermarket. How do you know it's really what it
Mm.
says it is on the label? Uh, the most common way of testing that is to extract DNA and do DNA sequencing.
Hmm.
But that's slow, that's expensive. You can't do it on site. So some colleagues of ours from Brandeis University developed a method where you can actually stab that fillet with a toothpick, mix a little test tube with that
Mm.
toothpick, and then in a 2-hour process, identify uh, that seafood item to species, probably for a cost around 10 to $15, something like that.
Wow.
So a much less expensive, more rapid process that can potentially be done right, on the dock or in the seafood warehouse.
Yeah.
Um,
Yeah.
So there are a number of interesting applications. I could go on, but I think that sort of illustrates
Now, that's a
the
wonderful
kinds of
overview.
applications.
Dan, what responsibilities come with safeguarding genetic resources for future generations?
Yeah, that is a huge it's a huge responsibility. Fortunately, it's shared by a variety of collections around the world. And, I think this is very important. Um, there's always a balance in everything we do between efficiency and redundancy, right?
Hmm.
Redundancy makes things safe because you have multiple copies. If one is lost, you still got the other one. But it's, of course, more expensive and less efficient to have multiple collections around the world. So we need to strike a balance between that. And the way we try to do that is through networks. We try to um, collaborate and align ourselves with various institutions around the world who are doing similar things or trying to do the same sorts of things that we are doing. So that helps to share the responsibility.
Hmm.
Um, but the responsibility goes on into many other areas that you might not think about until you try to begin a biorepository. For example, there's many legal and ethical aspects
Yeah.
of what we're trying to do. So back in the 90s, uh, there was a very famous convention on biological diversity. And that was an international agreement that where nearly all nations of the planet, the US accepted, agreed to consider the biological diversity of the planet um, part of the shared responsibility and shared property of the nations in which those organisms reside,
Hmm.
right? So it was an attempt to say, um, let's share the wealth, right? And
Yeah.
also the responsibility for preserving these organisms. Um, Many years later, like 2014, I think, that was followed on by something called the Nagoya Protocol, which took the next step of saying, okay, that was the overall concept. How do we apply that to things like DNA samples or DNA sequences? So the Nagoya Protocol attempted to establish a variety of principles that required repositories like us and researchers around the world um, to request permission from the nations and
Mm.
individuals and even like local communities where the materials are being collected
Hmm.
to create what we call access and benefit sharing agreements. So that, yes, if we discover something fantastic from these materials, some benefit needs to go back to the nations or the people
Hmm.
that helped us find those organisms. And also, typically those agreements go beyond just, dollars and cents sharing, but also kind of sharing knowledge, helping to train students in these various places. So the benefit goes beyond, just the new pharmaceutical drug.
Hmm. Wonderful, wonderful. Dan, what excites you the most about the future of marine genomics?
Wow, that's a great question, and one that's so wide open. Um, you know, to me, the most important thing is that we learn how to live with the ocean um,
Mm.
in a way that the ocean and marine environments and people benefit. We've managed that for the past how many millennia, only up until
Yeah.
essentially the last 100 years
Hmm.
has that started to become problematic. Um, and we're on a trajectory that's not good.
Right.
Right? We know this. We've known it for a very long time. It's very hard to get people to recognize this and to do the hard work that it takes
Yeah.
to make sure that we're living essentially, within our means. And I don't just mean financial means, but within our means to survive on the planet.
Yeah.
The oceans are incredibly important, right? Somewhere between 15 and 20% of the protein that humans rely on for food comes directly from the ocean.
Mm.
Un Untold millions of people rely on the oceans for jobs, for recreation, food sources. Um, the oceans also mediate just about every process on the planet, right? Most of our planet is ocean. So that
Sure.
means it's the biggest influence on our atmosphere. Half the oxygen we breathe comes from marine organisms, being produced by marine organisms. Um, it's terribly important that we learn to live in a sustainable way with the marine environment. Um, so far, technologies are helping, but not much, right? I mean, we're learning how to do things, um, but we haven't really shown the resolve of how to take what we're learning and put it into action.
So
What I'm
true. So
hoping
true.
with regard to marine genomics is that the most important thing we really need is to understand how the ocean works. Is to under Because how can we help? How can we protect what we don't understand, right? When we don't know what's going to happen if we do X, what Y will result from that?
Yeah.
We just don't know, and no amount of money or effort is going to help until we can understand the oceans. And that's where I think marine genomics is going to be fantastic. Um, every organism out there has a genome, to get a full sense of what is in the genome or how much information is in a genome. The genome of a single bacterium, which is 1,000 times smaller than a typical animal,
Mm.
uh, has about 1,000 pages worth of DNA text.
Wow.
If you were to write the text of the DNA out on 8 by 12 or 8.5 by 11 pages, it'd be about 1,000 pages of text. So multiply that by 1,000 and you get the amount of information that's typically in an animal genome. Um, that's phenomenal. Multiply that by the millions of species on this planet,
Wow.
and
Mind
that is
blowing.
an incredible resource of information that largely remains untapped.
Mhm.
But we're working very hard to move towards that goal of sequencing as many of those organisms as we can, and then the massive job of trying to figure out what all those strings of letters actually means in terms of biology and the real world.
Wow. Well, Dan, this has been fascinating. We could go on and on. There's so much more to cover, but unfortunately, we've come to the end of the program. How can viewers and others keep informed of what the center's involved with, possibly get in touch with you?
Sure. So we have a website, uh, which I can never remember the
We'll
actual.
put it in the credits. We'll have it
Yes.
in the
Okay.
credits.
We have a website that you can reach us at. We have a contact, oglinfo at northeastern.edu. Uh, and we're always eager to hear from people in the public. Um, People think that we're only interested in working with top scientists or, only interested in the most obscure and interesting organisms in the ocean. That's not true. We work with lay people all the time, people who are collecting various kinds of organisms for different purposes. We're always happy to talk to them to evaluate whether those materials can go into our collections. And very often they can.
Mm,
Uh,
nice,
so
nice.
we're always interested in outreach to the public and in the ways that public can work with us to help preserve
Terrific,
organisms.
terrific. And before we sign off, I also want to note that you've provided us with a wonderful video hearing from some of your other colleagues related to the center, which will be linked in our credits as well. Just want to close, Dan, with thanking you again and wishing you continued success with future endeavors.
Well, thank you for this opportunity to talk about our work.
Also want to thank those of you watching for joining us and hope you'll be able to tune in next time.